So thank you, Klaus, and thank you to all of the speakers for the outstanding presentations today. We have just a few minutes left, and I invited some of you to come and join a panel if you would. So could you move up to the front? I mean, we have a few questions. I think, I'm sure you have questions because we didn't ask any all day. But maybe we can reserve a few minutes now with our panel. And we got enough chairs for everybody? Good. So just make yourself comfortable here. I hope this is working, yes. So maybe we should ask a question, maybe Michael, you could start. I mean, we've heard about all the work with these software tool developers. Maybe there's some software tools that you would aspire to have. And also we heard about these repositories. How do you see the repositories having a role in your own work? And would it do anything to help facilitate your ability to work with some of the software tool developers?
So those are very loaded questions. So the first thing I would say is that I think having validated tools for deriving patient-specific measures is key. And there's a need for that because most of the way the tools are used, and I think a lot of what's already out there could be applied in this way, but the way many of the tools are used are to do these very sort of staid, group-wise comparisons, and as I tried to make the case this morning, we're probably missing a lot of information there. That's where potentially the repositories could come in and have a role. And the idea that, and I'm losing track now, but someone was talking about having controlled data in the cloud and having it accessible across institutions. If that can be done reliably, that would be an amazing step forward. But, you know, the realistic, realistically, are we going to get to a point where you can standardize to that extent across institutions? I don't know. And I think that tools that focus as a first step on locally validated normative data is probably the first step that needs to be taken.
Yeah. So, Timo, could I ask you, you have vast experience with, a number of the TMI imaging data sets. Are those data sets going to be in the INTIR INTBIR repository? Are you contemplating, making that contribution? And if so, can you tell us a little about how that's done?
Actually, I have experience only of tractography, so I cannot really answer very good to that question.
But you do tractography on TBI patients, as you showed us.
Excuse me, I didn't really understand the question.
Yes, so are you going to use this repository in your own work, the INTBIR?
Yes, I'm using tractography for the clinical diagnostics of TBI, but I have experience only of the Philips system. So
yes. Will you share your data with people at other institutions?
that there are many difficulties because their results depend on the system, on the DTI segments, on the machine you use, and also on the tractographic protocols. So it's very difficult to analyze images which are from another center or from another MRI unit. So I analyze only images of one MRI unit.
Yes, I see. So, Gary, can I ask you, for your work with TBI data, where does that data originate?
Actually, personally, I haven't worked with TBI data.
Oh.
But I think just add to actually the point Dr. Lipton was mentioning, is I think having a open public repository of data sets such as TBI or say ADNI or any other disease is tremendously valuable, especially for someone like me, for two reasons. One, obviously, for validating the tools I develop. That is important scientifically. But secondly, just as a way to disseminate my tool and to have available, say, to tell a user, here is a tutorial, and here's a set of data. You can actually follow a tutorial with real data and see what you produce. And I think that's tremendously valuable from a tutorial dissemination point of view.
Yeah. Olivier, can I ask you, I think you told us you're not working with TBI data today, and probably no one in the INRIA group, I would presume, is concentrating.
Yes.
So the question really is what would it take to attract workers from your groups to embrace TBI data and to begin to try and develop specific tools for that purpose? What could we do?
Well, from what I've seen today, I don't really know TBI. I didn't really know TBI before today. And
sure.
From what I've seen, it's really some studies that are quite close to other things that we're already doing on other topics. And I think it's just the opportunity that is missing, some collaboration, having some public data sets also is valuable. Because we've seen that INRIA before, I think in Asclepios, they were not doing anything on Alzheimer's until very recently. And since they had the opportunity to access the database, they just jumped onto the occasion, because we are more onto the side of developing some generic methods. And if we get the application, yes, we'll do it.
But what we've seen is the development of common data elements and the development of repositories, and now efforts to develop more standardization. And you could think of that as some one of the motivations is to try and attract the premier software tools developers to embrace those things. And I recognize that many people may not be familiar with the detailed needs of TBI data analysis, especially how they would differ from, the data sets that you work with every day. But perhaps this could be the basis for, establishing that dialogue. And but Lou, from a manufacturer's standpoint, you showed software tool development that was in a research laboratory of your own. Is it your expectation that would eventually go into the hands of clinical sites for testing, and how does that work?
Yes, eventually. I mean, at least within Philips Research, we develop tools and we disseminate these tools to different clinical sites that work with research to do clinical evaluation before they become products. So eventually the brain model that I presented today will be distributed to different clinical sites for them to do validation, and if data was available from a repository, I assume that they would be able to use it eventually. And personally, for me, I mean, it took me about 2 years to develop the initial version of the model, and most of that time it took just to create the training data. If there was a available repository with that data, I would be happy to use it. I mean, it would save me quite a bit of time, even though I work for an industrial company. It's still a repository will be useful. When it comes to traumatic brain injury in particular, to addressing the clinical needs of traumatic brain injury, I personally believe that there are questions that we need to, we definitely need more standardization, and we need to know exactly what we are looking for. And after that, people could develop tools, and we could use repositories to evaluate the performance of the tools and to come up with tools with the best accuracy possible.
Right. Michael, please.
One comment I just wanted to make is that a lot of what we're hearing is that it's extremely valuable to have this data available so that new tools can be applied to it. It can be used for training. It can be used for validation. And I think there's also new science that can clearly come out of that. If I do a study and I put data into FITBR, as I am, when that becomes available, there are things other people can analyze in that data that maybe I didn't even think of. But I think what we're also hearing, and this is a key point for me, is that when it comes to imaging data, and this is different from things like cognitive outcomes or even serum biomarkers, that there's so much variability in the data acquisition level alone that comparing quantitative diffusion measures from site to site is something that you can't really think about until it's been validated, and thinking about how you're going to validate that and whether it's going to work is a real question. So I just wanted to raise something that we've actually been thinking about a lot, which is there somehow a metric that could be standardized and disseminated? And I actually didn't show this in the data here today, but a predictor that we've been looking at is that in identifying areas of abnormality in an individual based on a comparison to a locally validated set of control data. So the actual diffusion metrics that come out of there, FA, RD, kurtosis, whatever it happens to be, are things that are very problematic in terms of pushing those across sites. But something what we've looked at as a predictor is simply the volume of tissue that meets the criteria for the abnormality. And that's just one example. I think in a similar sense, if you think about SMRI and structural imaging, so it's probably going to be a lot shorter timeline to generating volumetric measurements, as has been done in ADNI, for example. So I think we might need to think creatively about predictors where the initial identification of the abnormalities might be defined locally based on specific protocols that have been validated. But since some of the predictors might not generalize across sites, we could think about other types of predictors, a little bit out of the box perhaps, that might be generalizable.
Right, and another issue with doing multi-center clinical trials, maybe not so much with diffusion imaging, but in other types of imaging, is that you have a site qualification process and you have some sort of phantoms that have to be distributed, and you have to maintain the performance of the instruments within certain norms. Much of that seems to be non-existent, but maybe I'm wrong. Does anybody use phantoms for qualifying these systems, or
we're waiting for the phantom to be made.
Yeah.
So I just want to actually, I think we have here an expert on building phantoms.
Oh, great. Wonderful. So please tell us.
Thank you, Gary.
Tell us who you are, and then tell us a little bit about phantoms.
So yes, I mean, you're right. In diffusion imaging, there are not many phantoms around. And we have, a couple of years ago, thought about different ways to build phantoms. And in the beginning, we have recognized that there's a lot of interest. So we have kept developing those phantoms. And in the meantime, we have a really
nice construction process for such phantoms. So we take polyamide fibers and wind them in a semi-automatic process around spindles, put them into agar gel, and so we can send them around. We can build different types of fibers. We can build crossings. We can vary FA. We are working together with different collaborators that use those phantoms to do standardization in multicentric studies. We use those phantoms to validate tractography, to validate microstructural imaging. So there are many applications, and I think there's a lot of potential in building more elaborate phantoms in diffusion imaging.
Very important, very important.
I was just going to ask about the potential for 3D printing in the construction of.
So the problem in diffusion phantoms is that you need water. You need to include water somehow. So 3D printing would be thinkable for the raw types before winding those fibers. So this could be 3D printed. So we use our factory that builds those with micromillimeter accuracy, but you could also do that using 3D printing. We have thought about using 3D printing to print like pores of different shapes, but we actually haven't done it yet. So the problem is always you need to get water in there. So it's not enough to just leave room for the cells or for the pore size of pore shape, but you need to get some water into the phantom. So it needs to be porous.
Very good. So any last questions or comments from anyone? I see it as one of the challenges that we have is that, the important organizations have made an investment and commitment to these repositories. And, if I predict that they're going to eventually be asked what was the impact of this investment. And if you look at the autism community, they embraced this autism archive on which the Fitbeer was based. And it's very highly populated, and there are some publications which have come out which people in that community would say, were it not for the existence of the repository, these observations would not have been made. And I think we look forward to a time when the collaboration between the clinical users and developers of software tools in diffusion imaging and TBI work together and produce something, hopefully using these repositories that could not have been done by independent groups working separately. So with all of that, I want to thank our distinguished panel, and I hope that what happened today is just the beginning of cooperations between many of the groups here. Thank you all very much.
Thank you.